Chapter 13 of your Immunology textbook covers flow cytometry and laboratory automation. In this chapter, we'll cover flow cytometry, the instrumentation used, data acquisition and analysis, sample preparation, and clinical applications of flow cytometry. In addition, we'll touch on immunoassay automation in the clinical laboratory. Cell flow cytometry is an automated system in which single cells or beads in a fluid suspension are analyzed in terms of their intrinsic light scattering characteristics. The cells are simultaneously evaluated for their extrinsic properties, specifically the presence of specific surface or cytoplasmic proteins. Cell flow cytometry uses fluorochromes to identify cells. A fluorochrome or a fluorescent molecule is one that absorbs light across a spectrum of wavelengths and emits light of lower energy across a spectrum of longer wavelengths. Each fluorochrome has a distinctive spectral pattern of absorption and emission. By using laser light in flow cytometry, different populations of cells or particles can be analyzed and identified on the basis of their shape, size, and antigenic properties. Flow cytometry allows for rapid and accurate detection of cells found in small numbers in a sample. Flow cytometry is frequently used in leukemia and lymphoma diagnosis. In addition, flow cytometry has been used in functional assays for various diseases. It's been used to identify HIV infection and for immunophenotyping of cells. The major components of a flow cytometer include the fluidics, the laser light source, and the optics and photodetectors. The data analysis and management is done by a computer. For the cellular parameters to be accurately measured in the flow cytometer, it's important that cells pass through the laser one cell at a time, so cells are processed into a suspension. The cytometer then draws up the cell suspension and injects the sample inside a carrier stream of isotonic saline to form a laminar flow. The sample stream is constrained by the carrier stream and is thus hydrodynamically focused so that the cells pass single file through the intersection of the laser light source. And each time a cell passes in front of a laser beam, light is scattered and the interruption of the laser signal is recorded. As a result of the cells passing through the laser, light is scattered in many directions. The amount and type of light scatter can provide information about the cell's physical properties. So light at two specific angles is measured by the flow cytometer, the forward angle light scatter and the side scatter, which is also called the right angle light scatter. The forward angle scatter is considered an indicator of size, whereas the side scatter signal is indicative of granularity or the intracellular complexity of the cell. These two values are considered the intrinsic parameters and can be used to characterize different cell types. Unlike the forward scatter and side scatter, the extrinsic parameters require addition of a fluorescent probe for their detection. So fluorescent labeled antibodies bound to the cell can be detected by the laser. By using these fluorescent molecules with various emission wavelengths, one can simultaneously evaluate an individual cell for several extrinsic properties. The utility of these multicolor analysis is enhanced when the fluorescent data is analyzed in conjunction with the forward and side scatter. The combination of data then allows for characterization of cells according to size, granularity, DNA and RNA content, antigens, total protein, and cell receptors. Once the intrinsic and extrinsic cellular properties of many cells have been collected, and this is typically 10,000 to 20,000 events that are collected for each sample, the data can be digitalized, and then it's ready for analysis and evaluation by the operator. Each parameter can be analyzed independently or in any combination. Graphics of the data can be represented in multiple ways, and the first level of representation is a single parameter histogram, which is depicted on this figure on the slide. So the single parameter histogram plots a chosen parameter, which is generally fluorescence, on the x-axis versus the number of events on the y-axis. So this is why it's called a single parameter, because that's what's being analyzed in this type of graph. The operator can then set a marker to differentiate between cells that have low levels of fluorescence, which would be considered negative, from cells that have high levels of fluorescence, the positive cells, for a particular fluorochrome labeled antibody. The computer will then calculate the percentage of negative and positive events from the total number of events collected. The next level of representation is the bivariate histogram, or a dual parameter dot plot, where each dot represents an individual cell or event. Two parameters on each axis are plotted against each other. Each parameter to be analyzed is then determined by the operator. Using dual parameter dot plots, the operator can draw a gate around a population of interest and analyze various parameters, both extrinsic and intrinsic, of the cells contained within that gated region. This gating allows the operator to screen out debris and isolate subpopulations of cells of interest. Gates can be thought of as a set of filtering rules for analyzing a very large database, so the operator can basically filter the data in any way and set multiple or sequential filters or gates. The figure pictured here is an example of a dual parameter dot plot. Both parameters on the x and y axes are chosen by the operator, and in this case, lysed whole blood cells are analyzed on CD45, which is on the x-axis, and SSC on the y-axis. The operator then draws a gate or isolates the population of interest. So in this example, differentiating the granulocytes, monocytes, and lymphocytes for further analysis. When analyzing a population of cells using a dual parameter dot plot, the operator chooses which parameters to analyze on both the x and y axes. The operator divides the dot plot into 4 quadrants, separating the positives from the negatives in each axis. Quadrant 1 consists of cells that are positive for fluorescence on the y-axis and negative for fluorescence on the x. Quadrant 2 consists of cells that are positive for fluorescence on both x and y axes. Quadrant 3 consists of cells that are negative for fluorescence on both the x and y axes, and quadrant 4 consists of cells that are positive for fluorescence on the x and negative for fluorescence on the y-axis. The computer then calculates the percentage of cells in each quadrant based on the total number of events counted. So these figures show a quadrant analysis of a dual parameter dot plot. So again, on each axis, there are positive, which fluoresce, and negative, which are cells that do not fluoresce. In this example, on the right, they're trying to identify CD4 T cells. So CD3 is on the x-axis and CD4 is on the y-axis. So from what was just described, you can see that cells in quadrant 2 are positive for both CD3 and CD4. So these are the true CD4 T cells. Whole blood, bone marrow, and fluid aspirates are the samples that are commonly used for flow cytometry analysis. Whole blood should be collected in EDTA, the anticoagulant of choice for samples that are processed within 30 hours of collection. Heparin can also be used for whole blood and bone marrow and can provide stability in samples over 24 hours old. Blood should be stored at room temperature before processing and should be well mixed before being pipetted into staining tubes. Hemolyzed or clotted samples should be rejected. Peripheral blood, bone marrow, and other samples with large numbers of red blood cells require removal of their erythrocytes to allow for efficient analysis of white blood cells. Tissue specimens collected for analysis need to be transported in tissue culture medium at either room temperature or at 4 degrees Celsius if analysis is delayed. The specimen is then disaggregated to form a single cell suspension, and this can be done by either mechanical dissociation or enzymatic digestion. Mechanical disaggregation, or what's called teasing, is preferred, and that's accomplished by using a scalpel or forceps, a needle and syringe, or a wire mesh screen. Antibodies are then added to the resulting cellular preparation and processed for analysis. As we've seen before, the antibodies used for a lot of testing, including flow cytometry, are typically monoclonal, each one having a different fluorescent tag. Flow cytometry has become more routinely utilized in the clinical laboratory. Routine applications include immunophenotyping of peripheral blood lymphocytes, enumeration of CD34 stem cells in peripheral blood and bone marrow for use in stem cell transplantation, and immunophenotypic characterization of acute leukemias, non-Hodgkin lymphomas, and other lymphoproliferative disorders. Immunophenotyping by flow cytometry has become a very important component of initial evaluation and also subsequent post-therapeutic monitoring in leukemia and lymphoma management. White blood cell immunophenotyping is also essential when immunodeficiency is suspected. Enumeration of peripheral blood CD4 T cells in HIV-infected patients remains the highest volume test performed in the flow cytometry laboratory because it's used in classifying stages of HIV disease and determining treatment options. Flow cytometry is also the method of choice for the diagnosis of several inherited chronic diseases. With some of these X-linked and autosomal recessive diseases in which neutrophils are defective in their oxidative bursts, flow cytometry is used to expose the neutrophils to a dye which is not fluorescent until oxidized. When normal neutrophils are stimulated in vitro, the dye is oxidized and becomes intensely fluorescent. Neutrophils of patients with these inherited diseases are unable to oxidize the dye and do not become fluorescent after stimulation. And finally, one last important use for flow cytometry in clinical diagnosis is in cytometric bead arrays. So in this technology, various sizes and different fluorescent beads are used to determine multiple analytes at the same time. For example, 6 different sizes or colors of beads could be coated with 6 different autoantigens, and then patient sera is added, followed by fluorescent anti-IgG. If the patient has any autoantibodies, the respective bead will be fluorescent and can be identified and quantitated. So far we were talking about flow cytometry and that testing and instrumentation. So in addition to flow cytometry, the use of automated technology has become more prevalent in the clinical immunology laboratory with the advent of automated immunoassay analyzers. So this type of instrumentation has been available since the early 1990s. Currently, there's over more than 60 different automated immunoassay analyzers, and they're capable of performing almost all common diagnostic immunoassay testing, and they've largely replaced manual testing, especially in larger laboratories. We've even seen this here in our hospital where we had smaller instrumentation that we performed some of our immunoassays on that involved more manual manipulation. And these tests were added onto our larger automation, such as our Siemens Advia Centaur or Siemens Dimension Vista. So the driving motivation for the development of these immunoassay analyzers has been the need to create an automated system that's capable of reducing or eliminating the many manual tasks required to perform analytical procedures. So eliminating these multiple manual steps decreases the likelihood of error because the potential error caused by fatigue or erroneous sampling is reduced. Laboratory professionals are also trying to streamline test performance to reduce turnaround time and cost per test. So automation helps with this, and also in a lot of cases it's more accurate and precise compared with manual methods. Other potential benefits of immunoassay automation include the ability to provide more services with fewer staff, saving on controlled controls, duplicates, dilutions, and repeats, longer shelf life of reagents, and less disposal because of outdating, and also the potential for automation of sample delivery with barcodes for better sample identification. With immunoassay automation, there's currently two main types of analyzers on the market. There's batch analyzers and random access analyzers. Batch analyzers can examine multiple samples and provide access to the test samples for subsequent reaction mixtures, but these batch analyzers permit only one type of analysis at a time. So this can be considered a drawback. Stat samples can't be loaded randomly, and there can't be multiple analysis on any one sample. The other type of analyzer, the random access analyzer, is one in which a modular system is configured to measure numerous analytes from multiple samples. So in these analyzers, many test samples can be analyzed and a number of different tests can be performed on any one sample. These are, again, some concepts we discussed in general lab principles, but there's various stages and tasks within the analytical stage of immunoassay automation. So the tasks include introducing a sample, adding reagent, mixing the reagent sample, incubation, detecting, calculating, and reporting the readout or results. So all or some of these tasks may be automated on various immunoassay analyzers. So this concludes the chapter on flow cytometry and laboratory automation, but of note, the chapter also covers some concepts in laboratory instrument validation, which are concepts that we've seen and discussed in detail in general lab principles. So to summarize what we looked at in this chapter, flow cytometry identifies and enumerates various cell populations by measuring their properties while suspended in a moving fluid medium. Each cell or particle passes through single file, a laser light source, producing characteristic light patterns that are measured by multiple detectors for scattered light and fluorescent emissions. The scattered light in a forward direction is the measure of the cell size, whereas the side scatter determines the internal complexity or granularity of a cell. A single parameter histogram shows a chosen parameter on the x-axis versus the number of events on the y-axis. A dual parameter dot plot displays two parameters plotted against each other. Gating is used to block off a particular region of cells to be analyzed further. Flow cytometry is the most commonly used method for immunophenotyping of lymphoid and myeloid populations, and clinical immunology laboratories are now utilizing a variety of automated immunoassay analyzers in addition to flow cytometry for assessing various immunologic disorders and diseases.